Alright, why are we just seeing Esther's head? Because there's a delay, that's why. Hey everybody, what's up? Ready to learn about some CRISPRs today? That's what we're gonna do. Let me, alright, why are we just seeing Esther's head? Open up a, because there's a delay, that's why. Hey everybody, what's up? Ready to learn about some CRISPRs today? That's what we're gonna do. Let me, alright, why are we just seeing Esther's head? Open up a, hey Alexis, how's it going? That's why. Hey everybody, what's up? Ready to learn about some CRISPRs today? That's what we're gonna do. Let me, alright, why are we just seeing Esther's head? Open up a, hey Alexis, how's it going? That's why. Hey everybody, what's up? Ready to learn about some CRISPRs today? That's what we're gonna do. Let me, alright, why are we just seeing Esther's head? Open up a, hey Alexis, how's it going? Trying to find how I could pull up. Hey everybody, what's up? It always has issues with this when I try to pull up a window about some CRISPRs today to watch. That's what we're gonna do. While I draw. Why are we just seeing Esther's head? Open up a, hey Alexis. Why does it have all these weird suggestions? Hey everybody, it always has issues with this when I try to pull up a window. Weird suggestions. Just let me pull up paint. I just want to draw while I draw. Why does it have all these weird suggestions? It always has issues with this when I try to pull up a window. Weird suggestions. Just the whole display. Just let me pull up paint. It's always annoying, but it works. Why does it have all these weird suggestions? Letting the world see my computer display. I always get issues with this when I try to pull up a window. Weird suggestions. Display. Covering my face. It's always annoying. Alright, so today we're going to talk about some CRISPRs. Sound is repeating in an endless loop. Letting the world see my computer display. Alright, did I fix the sound? Is it better? Can you hear me? Let me see, what mic is it using? How about now? Anybody? How's the sound? Is it good? Now it's okay. Alright, great. Yeah, so today we're going to talk about CRISPR. Alright, can you see my display? Can I draw on this? CRISPR. So, the story of CRISPR. A long time ago, there was some bacteria that evolved. And that's their genome. And here's a little flagella. And the bacteria is just swimming around, going all these places, doing all these cool things. And then all of a sudden, there's these things called bacteriophages. They're like viruses for bacteria. they come and they inject their DNA inside the bacteria. And when they inject their DNA inside the bacteria lyse and die, but they also make more phages, and they make the things that the phage wants to do, so the phage goes on and lives. Bacteria then are in this constant battle. There's a war. All the bacteria is on this side, and they have little guns, and they're shooting poo, 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 poo. And the bacteriophages are on this side, and they're like, we're gonna get you. Yeah, you can't stop us. And they're shooting missiles. And the bacteria are like, we gotta do something to stop all these bacteriophages from killing us. This stuff is getting crazy. So bacteria, their immune system, evolves CRISPR. CRISPR stands for clustered regularly interspaced short palindromic. Anyway, it's stupid. Whatever. Nobody cares what CRISPR stands for because it doesn't really mean anything to most people, right? So, all right, how does this work? Click. Yes? Select. Delete. So the bacteria develop this immune system. And basically, what this immune system is this protein called Cas9. This thing called a guide RNA. Sometimes it's a gRNA. Sometimes it's a sgRNA. sgRNA is composed of a crRNA and a tracer RNA, so you might hear it referred to by those names. Yeah, all the naming is pretty stupid. I know what you're saying. You don't have to tell me. And sometimes it also requires a piece of template DNA. So what the bacteria did is in their genome, they stored kind of like memories of these viruses, and these memories were located in the guide RNA. This guide RNA then would match to the bacteriophage's genome, and when it matched the genome, the Cas9 enzyme would cut it. So it would chop it up. So whenever the viruses tried to inject their genome into the bacteria, they would just get chopped up. And then when they got chopped up, some of those pieces would get added to the bacteria's memory, their genome itself, and then they would be able to defend against more different types of viruses. So bacteria develop their own immune system. Now, what does that have to do with genetic engineering, right? Now, what we know about genetic engineering is that if you have is that if you have like a piece of DNA and then this DNA gets cut or broken in some way, right? DNA gets cut. What happens is that this DNA needs to get repaired. Now, when this DNA gets repaired, two things can happen. One thing is what they call non-homologous end joining. Now, what that means is that these two sides, where the cut is, are just reconnected by an enzyme. And when they're reconnected, they aren't always reconnected properly. When that happens, there's usually inserts an error. So if we have a gene right here, and this gene's being made, right? It'll get to this part where the cut happened and where the DNA isn't perfect because it wasn't repaired properly, and the gene won't really work anymore. Uh-oh. Right? So CRISPR is sometimes used to remove bad genes. So if, say, instead of this being a good gene, if instead it's a disease gene, right? Now imagine if it's a disease gene and you want to make this disease gene to stop working, what you can do is you can use CRISPR with non-homologous end joining to cut it, right? And then this disease gene, when it gets repaired, and it gets repaired with some errors in it, then this disease gene stops working. No work for me. Right? Now that is the most commonly used type of CRISPR. The second type of CRISPR is called homology directed repair. What does that mean? Well, homology is a word that just means descent through ancestry. So it means the sequences are related. So when you have a DNA sequence like this, and it gets cut, what the enzymes do that come in to fix this is they look for a repair template, right? Is there another copy of the genome around? Is there another copy of this gene around which I can use as a repair template for this broken gene? Well, if you flood the cell with a template, right? So you just flood the cell with a template that matches, has homology to the gene, what you can do is you can repair the gene and insert in something completely new, right? Homology directed repair, the efficiency of this is much, much, much lower than non-homologous end joining because you're not just cutting the DNA, you're also trying to insert a completely new thing, right? So it's used less often, and it's much, 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 much harder to do in humans. Oh, you can't see anything. Damn it. I just did all that and you couldn't even see anything? Fuck. My bad. All right. So we got our two types of CRISPR. I'll just go over this again really quick so you can see the drawing. So we have our non-homologous end joining, right? And that's when you have a piece of DNA that gets cut and then gets repaired and doesn't get repaired properly, right? So an enzyme comes in, tries to stitch this back together, but makes a mistake. That happens more often than we'd like to admit. And then you have your other process, which is homology-directed repair. And that's when the DNA gets cut and we put in a homologous piece of DNA. Homology, you remember, just means it matches. It's the same thing. It's a stupid science word. It just means it matches the other piece of DNA. So we flood the cell with our homologous DNA with something different. And this different thing right here gets added to the genome. The problem is it gets added with a very low frequency, all right? So even in the best cases, it's like 1% or less. So homology-directed repair, actually changing the genome of, say, like humans or something like that, is really hard. It generally can only be done when you're selecting for it. So like embryos, right? Because with embryos, you can try a bunch of different ones. Or if you're doing it in cell culture, so like in a petri plate, right? And so that's something that they do. It's called, right? So homology-directed repair is something that's good for editing organisms that you want to edit the embryo of. If you're editing humans or any other adult animals or anything like that, the only type of genome repair you'd ever try is non-homologous end joining. So what that means is you can really only edit disease genes. You can't edit anything else because the only thing you can do is knock out the function of a bad gene. You can't change the DNA of a good gene to make it better, right? Or change the DNA of a gene to make it better. You can only make a gene not work, right? So that really limits the usefulness of CRISPR. All right, good. So it looks like we're all seeing this all now. So these are the two types of CRISPR. Now, in the majority of cases in real life, CRISPR is way overhyped, right? Because it's so limited in its use cases. So even if, say, you have a copy of a bad gene, the majority of what scientists do, so you have a bad gene inside you. Does that look like a bad, we'll make it like a vampire or something. It's a bad gene. So when you have a bad gene in your body, what it does is, remember we learned that DNA makes protein? So the protein it makes is it's defective. So it doesn't do the thing it's supposed to do. So if it was an enzyme and it's supposed to, cause a reaction, it doesn't. And doesn't. So the easy way to fix this is just to, you know, if we got a human being, is just to take a functional copy and stick it in this guy, right? So all we want to do then is just get rid of this guy, put in a new copy that is functional, and the old one doesn't really matter. It's not doing anything anyway, so it doesn't really matter. Now this is called gene therapy, right? Gene therapy. Now with gene therapies that have been approved today, what they do is they just take a functional copy of the gene, right? They put it in a circular piece of DNA called a plasmid that we're going to use, we've already used in this class, and they insert it into a human either using a virus to carry it in or just by injecting the DNA. You can literally just inject DNA, inject that into a human, and it will incorporate into cells. That's pretty crazy, right? Yeah. So in the majority of cases, even when somebody has a disease gene, you wouldn't want to use CRISPR. You'd most likely use gene therapy, right? So there's a lot of hype around this CRISPR thing. It's probably not going to get used as much as people imagine it will. You know? All right. No questions. Good. Feel free to ask questions. I'm listening. I'm paying attention. So in this class, we're going to learn to use CRISPR. The experiment that we are doing is in bacteria. Now, the majority of genetic engineering experiments are in bacteria because they grow fast, right? So you always want to use bacteria as your proof of concept. So the doubling time of E. coli is like 20 to 40 minutes, right? Now, compare that to like other cells, plants, right? In order to grow a plant, what are you talking? Even Arabidopsis, you're talking what, like 30 days? Now, human cell lines in cell culture, they grow much faster, right? So whereas you could see the results of an E. coli experiment in like 12 hours or less, cell culture experiment, you're probably talking 48 hours plus. Also, human and mammal cell culture, they're much more difficult to do because the cells are more finicky. They like really specific conditions, right? So they like very specific media. Usually we use FBS, fetal bovine serum, which is literally the serum. So the white blood cell, the non-red blood cell part of blood, the serum, spin down all the red blood cells and all the cells, the serum of fetal calves. So fetal bovine serum, right? Fetal cows. So they literally extract blood from fetal calves and use it to feed human cells. And that is the main source of growth media for human and mammalian cell culture. Pretty crazy, right? So Alexis Ortega says, can we use 3 viral vectors to cut the DNA in 2 places? Yeah, so what you can do, back to CRISPR real quick. What you can do is if you have a cell and you just have the Cas9 enzyme being expressed, you can add as many guide RNAs as you want that target, you know, all different genes in the cell, right? So, you know, there was a paper out recently by George Church's lab, and they claimed they did like 10,000 or some ridiculous number. But the problem was it was like repeats, so it was all similar places in the genome that had the same sequence. But people, you know, have done pretty high numbers. You know, like 10 or 20 different places in the genome they've been able to edit using a bunch of different guide RNAs. So if you wanted to edit something, all you'd do is you'd put in a bunch of different guide RNAs, right? You could also put in a bunch of templates, but the templates, right, are really low efficiency. So the chances that you get more than one edit in a cell successfully is probably going to be pretty small. All right. Any other questions? Looks like we're doing good. All right. So the experiment for the class is editing bacteria again. Now, remember, so if we wanted to insert a gene, say, into bacteria like GFP, it would be pretty big, right? So you got your promoter, you got your GFP gene, you got your terminator, and you're talking something about 1 kb, 1,000 base pairs to 2 kb, right? It's pretty big. And to make this GFP plus promoter plus terminator, you can't really get it synthesized. Right now it costs, to synthesize something that big in a reasonable quantity, you know, enough for one experiment would probably cost you 150 bucks. So you can't really do that. So how do you see the results of a CRISPR experiment then? Well, the best way to do that is a live dead assay. Live dead, right? Scientists have been using this for years. If the bacteria lived, it worked. If they died, it didn't work. So we know of a mutation in bacteria in this gene RPSL. Now RPSL is part of the ribosome. Now the ribosome is a giant protein complex that what it does is it makes proteins, right? So, well, it's RNA protein complex, but so what happens is your mRNA comes to the ribosome and out on the other side creates a protein, right? So RPSL is a gene that's part of this giant ribosomal complex. Now, we know that the antibiotic streptomycin, what it does is it binds to this RPSL gene in this ribosome complex. And when it does that, it prevents proteins from being made. No proteins get made. But if you mutate a specific amino acid in the RPSL, so it's amino acid lysine 43K to threonine T, then this streptomycin can't bind to the RPSL because lysine, the amino acid, is positively charged and threonine is negatively charged, right? So if you switch the charge, the way it was binding will be it won't bind at all. So this ribosome won't get inhibited, and what we'll see is we'll actually get protein being made. So the goal of the CRISPR experiment is we want to make a genetic mutation in a single base pair, which will change this amino acid residue in the RPSL protein. When that happens, the bacteria will live on streptomycin, the ones that are edited, and the ones that aren't edited will be dead. D-E-D. Dead. Does that make sense? Maybe? Yes, no? Maybe so? So your goal, if you choose to accept it, mission, right? Your mission, not goal. Your mission, if you choose to accept it, is to genetically modify the RPSL gene in this E. coli. Now, this should teach you about CRISPR, how it works, right? With the Cas9, gRNA, and template, the 3 different components. It should teach you how to do transformations, right? Antibiotic selections, all the things you need to know to be a genetic engineer. Now, after you finish this experiment, you might be like, okay, now how do I figure out if I want to design my own guide RNA, how do I do that? That's a good question. I'm going to show you how, right? So, all right. So now there's a couple websites you can use. So the first website is Chop. Chop Bar in San Francisco. What's going on? I'm disappointed. So here's the website address. Chop CRISPR. I'll post it in the classroom. Right there. If you can't see it, make sure you switch from top chat to live chat. All right? So Chop. Now, Chop is one of many websites that what it does is it allows you to pick the best guide RNA to target a gene you're trying to knock out, right? So this is what it tells you. You can even choose. What do I want? Knockout, knock in, whatever. We want knockout. That's the easiest. That's what we're going to do. There's all these different things, but we just want Cas9. Don't worry about the other ones. And now target. So anybody got a target gene that we want to look at? Well, you can look at a lot of genes. Let's say what gene has gene for, drinking all these beers has maybe put on a little weight. So what we could try to do is, what is a gene that's related to obesity? Let's see, obesity. We want to knock out that gene that's related to obesity. All right, let's try to find something that's recent. Oh, here we go. That's the FTO gene. What is this? The FTO gene leads the way. All right. So that's what we want to do, is we want to knock out this FTO gene. So that, yeah, we don't gain weight. Now, obviously, it's more complex than this. But it says right here, analysis of the FTO gene in mice showed loss of function is associated with no difference in energy intake but greater energy expenditure. All right. So we'll just assume in this case that if our FTO gene is not functioning, it's going to help us lose weight. So let's find out a guide RNA to knock out our FTO gene. Our rank is 2 in the queue. Looks like they have a lot of misspellings in the calculal mechanism coup. All right, come on. I'm ranked number 2 in the queue. Let's go. Somebody else beat me to it. Yeah. Thanks again. Oh, come on. All right. What's taking so long? Well, if you got a beer, you know, just hang out and relax for a minute while we wait to see what our results are. Uh-oh, I'm 1 in the queue. Here we go. I'm excited. I don't think this website should use such processing power. Like, what's going on? Am I missing something? Should take like 10 seconds. All right. So here we go. Now, what we get is we get a rank of gRNA target sequences. Oh no, what happened? I clicked on something. And we get a quote-unquote efficiency, we get a self-complementarity, and we get, you know, how well it matches. So we get whether it matches with exactly, it matches with 1 change, it matches with 2 changes, it matches with 3 changes, and how often in the genome. Now, obviously, this is just a computational thing, right? It's not 100% accurate. It's not even 90% accurate, right? You're talking, it's just a good guess at what would work. So everything that's on here would need to be obviously validated, right? But if it was me and I'm looking at this and I'm trying to knock out the FTO gene, your best choice is to start with number 1. It has the highest efficiency, predicted efficiency, and has some of the lowest matches in the genome, right? So it's probably a good idea to start with that. Generally, you probably start with a couple of them and see which ones work best, right? So chop is one of the major ones that is used, but there's also companies like Atom that will design gRNAs for you. All right, so what we want to do is we want to design our own gRNA. We want to use wild type Cas9. And let's try that again. FTO gene. And let's see what this one picks. So this one has CCA-GGCT. Nope. Let's see if it's even on. Let's see how the matches compare. Let's go Control C, Control F, V. Oh man. So the match from Atom doesn't even show up in the chop match. Oh, oops, maybe I included the PAM sequence. Still doesn't show up. All right, let's try this one. Dang, that one doesn't show up either. Interesting. So these two are predicting the same gene. There's no similarities between the two in terms of gRNA. Now, I don't know which one I would trust more. Yeah, so this is a company. So generally, I don't know, I would trust somebody who's trying to make money off me more than somebody who's doing something for free, because scientists aren't good at keeping up with everything because they can't publish, right? Another company that does stuff is Synthego, but their website is always broken. I don't think they actually sell anything. Let's see. Bioinformatics. Top scoring guide RNAs. Let's try it out. All right, let's select a genome. Let's go Homo sapien. Let's try FTO gene. They found 78 targets. Ah, man. So they actually write it in RNA. So remember, RNA has uracil instead of thymine. So they put U instead of T, which makes it difficult to compare. We can try a little bit. So that would be ATG, CTT, GT. All right, let's see if that matches anywhere in chop. Nope, nothing on chop. Nothing. Wow, so we got 3 predictions, and none of the predictions match between all 3. Let's try one more. TGG, CT, GC. All right, I have a feeling. Oh, nope. All right, wow, that's weird. That's interesting. none of them match. So we got a lot of different predictions. Yeah, I mean, there should be an ensemble site, right? So, yeah, so when you're choosing, say you wanted to design a CRISPR experiment of your own for any organism, humans, bacteria, yeast, anything like that, you know, which website you use is up to you. I'm sure there are people who swear by all of them, and I'm sure all of them have some amount of functionality. So usually the best idea and what scientists do is they choose a couple, maybe choose one from each website, I don't know, and see which one has the highest efficiency, right? So if you're doing an experiment in bacteria, right, a live dead assay, say, you would see which one at the end of the experiment has the most bacteria alive. Makes sense, right? And which one has, bacteria dead. So that's that. All right, so do we have any questions about this CRISPR stuff right now? Anything you want to know? I'm here to answer your questions. Um, I, I hate how YouTube is always delayed. So the video is delayed by like 10 seconds, and then the responses are delayed by like another 10 seconds. So there's like 20, 30 seconds between any questions. God damn you, YouTube. How can you imagine improving the HDR method? So there's a lot of things people have tried, but you have to understand that on a scale like a whole human, it becomes very difficult. So there is a paper let me find it really quick, in which they use a technique that's not CRISPR, right? So you can find a link here. So what this is it's basically just a virus. And what the virus does is um, the virus does the insertion of the gene into the genome. So it does essentially homologous recombination or homology directed repair or whatever you want to call it. And it doesn't require a template, or the template is just included, right? You just put in a template with sides, the homology sides that match, and it'll insert it into the genome without having to cut the DNA or anything like that, right? The main problem with the probabilities is that the probabilities go down every time you have to do something, right? So first, you have to get DNA inside the cell. And go ahead and ask your question, Kent, whenever you're ready. So you have to get the DNA inside the cell. That's the first thing. The second thing is, once you get the DNA inside the cell, then you have to get the CRISPR, you have to get the Cas9 or guide RNA components made if they're not already made. You have to get them inside the nucleus. Once they're inside the nucleus, then you have to find the correct place in the DNA and cut it. Right? So already it's difficult. Now, after that, if you're trying to do homology-directed repair, then the enzymes that repair the genome, what they have to do is they then have to find the template. The template also has to get into the nucleus, and then they have to, recombination has to happen properly, right? So you're talking, once you get down to that final probability of everything working, it starts to get really small. Alexis, if you can't see the link I posted, switch from top chat to live chat in the top left corner of the chat window on YouTube. Switch it to live chat and you should be able to see the link I posted in this paper. All right, Kent. Now Kent's question. So since a likely nucleotide sequence can reverse translate from a protein sequence, wouldn't it be possible to make plasmids that emulate expression of extinct animal proteins described in literature? Yeah, I mean, you totally can make extinct animal proteins, right? That's not a big deal at all. You can put them in plasmids or do whatever. I think the big problem is that DNA gets degraded. So the half-life of DNA, spontaneous, like bond breaking, is about 50,000 years. So that means if you're an organism that was alive 50,000 years ago, half your DNA is probably gone, right? At least. And all the rest of your DNA is chopped up in an unbelievable rate, right? That's not good. So when we're trying to find out the DNA or protein sequences of extinct species, we generally have a really, really, really hard time. Really hard. And so there's not much of that out there, actually. Fluorescent proteins in the skin rather than tattoo dye. That's good. That's a good idea. The one problem with that is just getting the fluorescent proteins to express at a high enough level that you can see it with your eyes, right? So it's easy to get DNA in cells. But the problem isn't just getting DNA in cells, it is getting it in enough cells so that you see the outcome that you want, right? All right. Any other questions? What we got? Yeah, just constantly waiting for this lag to catch up. I think for the next class, we're going to switch to another platform. Find some platform that people can respond instantaneously. Yes, Mr. Metric asks, does being frozen reduce DNA's degradation rate? Yes, so chemical reactions and enzymatic rates are reduced by temperature, right? So theoretically, at absolute zero, atoms are not moving at all. So the degradation rate would be 0. The closer you get to absolute 0, the slower the rate of the atoms are going to move, the slower the rate of chemical reactions, the slower the rate of degradation. That's why we tell people to store your DNA in the freezer. It's not that if you don't do that, the DNA is all gonna go bad. It'll, probably be fine. But because it slows down all the enzymatic rates and it slows down all the chemical reactions, the chances of it being bad are much, much, much lower than if you just left it out at room temperature. Right? So that's generally the case. You want to store DNA in general at a colder temperature. It just helps prevent it from being degraded. All right. All right. We don't have any more questions, then thank you for coming to class today. I appreciate it, everybody. If you have questions, feel free to post them in the classroom and we'll do our best to get to you in a reasonable time, but also understand that we can't always answer your questions right away. Appreciate the patience, and get doing your CRISPR experiment and let us know how it goes. All right. I will talk to you all next week.